Hello everyone, I am SARITA RAWAT, Assistant Professor, Suresh Gyan, Vihar University, Jaipur, School of Pharmacy. Today we will start our next topic of pharmaceutical microbiology, stain and staining techniques. So, learning outcomes of today's lecture included you are able to know about the staining techniques, type of staining techniques, importance of staining techniques, staining methods. Objective included today's lecture are you are able to know about the visibility by great contrast between the organism and the background, how it employs. Then you are able to differentiate various morphological type and size arrangement of bacteria, virus, any microorganism. Then you are able to determine the staining characteristics of organism and at times direct diagnostic of disease and demonstrate the purity of culture. And you are able to observe certain structure like flagella, capsule, endospores, etc. So these are the objective of staining techniques. Now we will start with the introduction part. So, bacteria are microscopic organisms. They are also colorless for the most part. In order to visualize them, to study their structure, shape, and other structural characteristics, it becomes necessary to make them more easily visible. This means that the structure have to be constructed from their environment so that they can be seen easily. Now, what are stains and what are dyes? So, when we talk about the dye, a dye is a general purpose coloring agent, whereas a stain is used for coloring biological materials. So, we can say if we want to color any agent and if we want to know about any material, so we have to use the coloring agent. Now, a stain is an organic compound containing a benzene ring plus a chromophore and an oxochrome group. Chromophore is a chemical group that impacts color to the benzene. And oxochrome group is a chemical compound that conveys the properly or property of ionization of chromogen, that is ability, it has a ability to form salt and it bind to the fiber or tissues. That's how they give color to the particular biological material and they are easily available to visualize. Now we have a types of dye. So when we see about the types, we have acidic, basic, and natural. And while we're talking about the acidic, we have negatively charged acidic radicals impacts color in the eogens, acid fusions, and we have marichite green, nigrosin dye, and Indian ink. These are easily available in any laboratories. Now we have basic dyes. Basic dyes are positively charged bases radicals that combines with the negatively charged particle in cytoplasm and give color. For example, we have methylene blue. This is very basic dye we used in our day-to-day laboratory practices. Then we have crystal violet, janitin violet. Now, coming to the natural dye, we have both positive. They have both positively and negatively charged impacts. They differentiate colors to different components. For example, we have Leishman stain, Wright stains, and Garimas stains. So these are the type of stains dye we have. Now, what are the staining methods we have? So talking about the positive staining, where the actual cell are themselves color and appear in a clear background. So we have two kinds of positive staining. The first one is simple staining. So simple staining, a stain which provides color contrast but gives same color to all bacteria and cells. For example, Lofier's methylene blue, polychrome methylene blue, dilated carbon fusion. Next, we have differential staining. Differential staining, a stain which imparts different color to different bacteria, is called differential stain, which contain more than one stain. For example, we have gram stains, acid fast stain, and special stain. Coming to the next part, we have negative staining. Negative staining is where the cell remain colored, uncolored, and the background is colored to create a contrast to add in the better visualization of the image. For example, we have Indian ink and nigrosin ink. Now, bacterial smear preparation. How we can prepare a smear to check the morphology of any bacteria, virus, or any microorganism. So first we have to prepare a smear. The visualization also depends upon the type of smear we are preparing. So smear is a distribution of bacterial cell on a slide for the purpose of viewing them under the microscope. And the method include of making a smear. We have to aseptically a small sample of the culture is spreaded over a slide surface. So we have to take a slide, then we have to take aseptically small sample over the slide. We have to spare it, then this is then allowed it to air dry. The next step is heat fixation to help the cell adhere to the slide surface. Then the smear is now ready for staining. So this is the method of preparing stain. So you can see in this picture, first we are putting our smear on the slide, then we are spreading it, then we are air drying it, and this is our final smear. Now, smear fixation. How can we fix our smear? So, first is our heat fixation. This we can done by pass air dried smear through a flame 2 or 3 times. Do not overheat. So we have to pass our slide over the heat 2 to 3 times so that it can be dry. We do not have to overheat it, otherwise it can break. And allow slide to cool before staining. Then after we have to cool it. Then methanol fixation. The second method is methanol fixation. So how can we do this? We have to place air dried smear in a coupling jar with methanol for 1 minute. Alternatively, food smear with methanol for 1 minute. So we have to do it, we have to float the smear with the methanol for 1 minute. Then we have to drain the slide and allow to dry it before staining. Now, requirements for staining. What we need to, what are the requirements we have to need before for staining? Okay, so staining, majority of the stain used for staining bacteria are of the basic types, as nucleic acid of bacterial cell attract the positive ions. Example like methylene blue, we have crystal violet, acidic stains, and they are used for the background staining. Then we have moderate stain. It is a chemical that forms an insoluble complex with the stain and fixes it or causes the stain to be penetrate more deeply into the cell. These are used in the indirect staining, for example, gram iodine in gram staining and phenol in Ziehl-Neelsen staining. Then we need acunators. Acuntators are, it is a chemical which when added to a stain to make the reactions more selective and intense. For example, potassium hydroxide add in the loeffler's methylene blue. So acantus is also important. Then we have a decolorizer. Decolorizer is used. It is a chemical used to remove the excess stain in the indirect regressive staining. For example, ethanol or gram staining. Now, these are the types of staining techniques. So, as you can see, we have categorized into 2 categories. And the first one is simple staining. Simple staining is used of a single stain, and then we have a differential staining. It is used to two contrasting stains. The first one, simple staining. Now we have categorized them into two types: direct and indirect. Direct is positive and indirect is negative. Now differential staining is of two types: separation into two groups. First one is gram stain and acid fast stain. Then the second one is visualization of staining. So for that we can see flagella stain, capsule stain and spore stain. Now we will see in detail about these staining techniques. So first we will start with simple staining. Simple staining, a staining method that used only a single dye, that which does not differentiate between different type of organisms. There is only a single staining step and everything is stained with the same color. Simple stain are used to stain whole cell or to stain specific cellular components. Type of simple staining: direct positive staining, we have staining object, and then indirect negative stain, it is stained to the background. So the difference between direct and indirect is only that direct is positive staining and it stain the object, and the indirect is negative staining and it stain the background. So bacteria have a site minus charges. So you can see basic dye color protection is positive. So it can use as a direct stain. So it will retain color. Then the acidic dye color portion is minus. So it will like show indirect skin. Now, direct staining is a positive staining, a simple staining technique that stains the bacterial cell in a single color. So, as you can see, the bacteria is in single stain, and there is no background staining. So many of the bacterial stain are basic chemicals. These base dyes react with the negatively charged particle cytoplasm. Opposite charges attract and then organism become directly stained. So for example, methylene blue, crystal violet and basic fusin. Now, negative staining. In this staining process, instead of cell background is stained, and here an acidic dye like nitrosin or Indian ink is used. Acidic stain carries a negative charge and repelled by the bacteria, which also carry a negative charge on their surface. Hence, an acidic dye do not stain the bacteria. So, as you can see in this picture, there is no staining in the bacteria, but the background is stained. Because of that, we are able to know about the bacteria shape, size. So instead, it forms a deposit around the organism, leaving the organism itself colorless or transparent upon examination. Now, staining general techniques, you can see we have to prepare the smear, then we have to air dry it, then we have to heat fix it, then this is the staining, and then we can look it over here. Now, importance of fixing the smear. Why we need to fix the smear? So, fixation accomplish three things, like it kills the organism, it causes the organism to adhere to the slide, and it attracts the organisms so that they more rapidly attract they more rapidly accept stains. Now, differential staining, gamma staining. Gamma stain. In the late 18th, Christian Gamma observed that some journal of bacteria retained a diiodide complex when rinsed with alcohol, while another genera were easily decolorized with alcohol, and it could be then visualized by a constructing container stain. So, It is decolorized with the alcohol and would be then visualized by a contrasting counter stain. So this stain procedure defines two bacterial groups, like those which retain the primary dye that are positive by the Gram's method or gram positive, and those which are easily decolorized negative by the Gram method or gram negative. So this is the staining point for the bacterial identification procedure. Now, gram staining, the difference is dye retention is dependent on such physical properties as thickness, density, porosity, and integrity of the bacterial cell wall, as well as its chemical composition. Now, gram-positive bacteria have thick, dense, relatively non-porous walls, while gram-negative bacteria has thin walls surrounded by the lipid-rich membranes. So, some non-bacterial organisms with thick cell wall, for example, some yeast, also stain gram-positive. Gram-positive bacteria, which have a lost wall integrity through aging or physical or chemical damage, may stain gram-negative staining. So, as you can see in this picture, there is a cytoplasmic membrane, lipotrypsic membrane, peptidoglycan, triconic acid. So you can see this is the gram positive staining and this is the gram negative staining. So these are the picture of cytoplasm and this is the plasmodic space. You can see this is the outer membrane. Now the dye, this is the dye going the inner part. So the mechanism behind the gram positive or gram negative staining. So, gram staining requirement. Gram stain is a 4-part procedure. The specimen is mounted and heat fixed on a slide before proceeding to stain it. The reagent requirement are crystal violet, the primary stain, iodine solution, the moderate stain, decolorizer, we need ethanol, saffron, the counter stain, and water, preferably in a squid bottle. So for gram staining, we need these our indicators, and then we need water also. So the whole process is very lethal and time consuming. Now, the procedure of gram staining. So first, the bacteria are first stained with the basic dye crystal violet, that is our primary stain. Both gram positive and gram negative bacteria become directly stained and appear purple after this step. Then the bacteria are then treated with the gram iodide solution, that is our moderate stain. This allows the stain to be retained better by forming an insoluble crystal violet iodine complex called an iodine lake. Then both gram positive and gram negative bacteria remain purple after this step. Then the third step is gram decolorizer. A mixture of ethyl alcohol and acetone is then added, and this is the differential step. Then gram-positive bacteria retain the crystal violet iodine complex, while gram-negative bacteria are decolorized. So as you can see, in the first step, both the bacteria are colored, and in second step, we are just fixing their coloration, and in the third step, while we are using the decolorizer, only the gram positive bacteria retain the same color and the gram negative bacteria will be decolorized. So finally, the counter stain saffron, also a basic dye, is applied since the gram positive bacteria are already stained in purple color, so they are not able to retain the saffron color, or we can say counter stain. Only the gram negative bacteria that are now colorless become directly stained by the saffron, and the gram positive appear purple and the gram negative appear pink. So it is very easy to differentiate between them now. So as you can see, this is the gram positive bacteria and gram negative bacteria. So while we are as the starting they are same, then we are heat fixation on them slide, then we are taking crystal violet, so stain purple, they are in first step, they both are in the purple color. Then in the second step, when we are using modern stain iodine solution, they are still in the same color. Then in the third step, when we are using ethanol as a decolorizer, so gram positive bacteria is still in the purple color, but in the gram negative bacteria, they are already decolorized with the help of ethanol. So now in the fourth and final step, we use the counter stain saffron, so it will color the gram negative bacteria, those who are already decolorized by the ethanol. So now we are able to see gram positive and gram negative bacteria separately. So this is the pictorial view of gram positive and gram negative bacteria after staining. Now the next staining technique, we have acid fast staining. So acid fast staining is basically structure of an acid fast cell wall. You can see it here. There are free myclonic acid and polypeptides. There are also the cytoplasmic membrane. So now acid fast staining, what is the theory behind acid fast staining? So when we see one stain, the acid fast bacterial cell resists decolorization with the acidified organic solvent, example acid alcohol and alcohol. So we called it acid fast. And acid fast staining property of the genus mycobacteria depends upon their lipid-rich walls, which are relatively impermeable to the various basic dye. So unless the dye are combined with the phenol, the exact method by which the stain is retained is unclear, but it is thought that some of the stain become tripped while the cell and some form a complex with the myclonic acid. So this is supposed by the finding that shorter chain myclonic acid or mycobacterial cell which disrupt cell wall stain weakly acid fast while we are using Nocrita. For example, in Nocrita. So this is the acid fast staining. As you can see, the dye turn out the bacteria into the violet color. So acid fast staining of Mycobacterium tuberculosis in sputum. So radish acid fast bacteria among the blue normal flora and white blood cells in the sputum that are not acid fast. So as you can see, these the bluish are the normal flora and these reddish are the acid fast bacilli, and the white part is our blood cells and sputum. Now we have another method of staining, that is spatial staining, or we also in spatial staining we will see capsule staining. So capsule staining is a diagnostically useful since it is a virulent factor. It is used in the pneumococci. Then bacterial capsule are non-ionic or neither acidic or basic stain will adhere to their surface. So the bacteria, those who are having non-ionic surface and non-acidic surface, then we use the capsule staining to identify these kind of bacteria. So capsule are demonstrated either by negative staining, Nigrosan or Indian ink, or by spatial staining, his method and Anthony's method. Now, what is his method? So his method, the capsule is non-ionic in nature, so it doesn't get stained by acidic stain, but a basic stain such as a crystal violet stain the cell as well as the capsule. So this is followed by the treatment with the hypertonic solution, 20% copper sulfate solution, which serve dual role of both the decolorizing agent and counter stain. So copper sulfate solution being hypertonic capsules, diffusion of the stains toward outer surface of the cell. After dyeing of the slide, the stain which is not passed from the capsule layer during diffusion retains in the capsular layer and copper sulfate thin decolorize the capsule. So capsule appear as a faint blue halo around a purple cell. So this is the Hess method of capsule staining. You can see it in this flowchart. Prepare a smear, air dry it and do not heat fix it. Flood the smear with the crystal violet for 4 to 5 minutes. Then rinse the slide with 20% of sulfate solution. Then we have to air dry the slide and observe it under the oil emulsion lenses. So you can see this. Now, next method we have endospore staining. So endospore staining. When we see endospore staining, we find out spores are normally impervious to stain. Then, under the light microscope, endospores have a high light refractivity indicative of high protein content. So, endospores can be stained by modified Ziehl-Neelsen method by using 0.25 to 0.5% of sulfuric acid as a decolorizing agent. Then we can also do this endospore staining by using Bartholomew Matters method. Then the another techniques we have for endospore staining is Sacrifier Floaton stain techniques. Now, what is Sacrifier Floaton method? So when we see the Sacrifier Floaton method, we need a malignant green is used to stain the endospore, that is our primary dye or primary stain. The malignant green is forced to permeate the spores wall by heating or moderate. Then we apply a moderate heating while we applying a malignant green because it permeates the spores. Then we wash it with water and remove the staining from the vegetative cells but not from the spore wall. So the endospores thus retain the primary dye, and while the vegetative cells they lose the primary stain and take the red color of the secondary stain saffron. Okay, so this is the method for Sacrifier Floaton method for the staining of spores by using malignant green and the secondary stain saffron. So this is the endospore stain of Bacillus magnetorum. So green endospore with pink bacilli. So these green endospores with pink bacilli we can see easily. So this is about the endospore staining. Now this is all about your staining techniques, and now we have some questions regarding this lecture. You can easily answer them while going through this lecture. So the question contains, what is the purpose of fixation in the smear preparation? Next, in the gram staining, which of the reagent actually differentiate between gram positive and gram negative cells? Now, what stain is used for visualizing mycobacterium? So we have already studied all these questions. While you will go through the previous slides, you can easily find out these questions answer. Which the cocci which mostly occur in single or pair are? Then which of the step reagent above distinguish between gram positive and gram negative cells? What cell structure is responsible for the difference in the staining deducted by the gram stain procedure. So next question. What would a smear of a mixture of gram negative and gram positive organism look like? And if the final step were optimated. So you can answer this. What is the purpose of doing a differential stain? What is the difference between a differential stain and a simple stain? Then the next question, why is the gram staining called a differential stain? So as you know, we have already studied these all topics in depth about all these questions. So we will while you are going through the previous slides, you will already know about these questions answer. So we will revise once again all the slides so that you can know about the questions I have asked to you. So this is the introduction of bacteria. Then what is stains and dye, why we used it. So this is the type of stains, acidic stain, basic stain and natural stain. Then these are the staining methods. We have positive staining, which includes simple staining, differential staining. Then we have negative staining. Negative staining include India ink and necrosin. Then we have bacterial smear preparation. So how can we prepare the smear? What are the method? How the method of preparing a smear? What is smear? So this is the smear preparation, the picture view of our laboratory. So this is the slide and we are putting a bacterial sample. Then we are just spreading it. Then we are air drying it. Then this is the final smear we have. Now, how can we fix smear? So we have a method, heat fixation, methanol fixation, and requirement for staining. So what are the basic requirement of staining? So we need a stain, a moderate stain, a quencher, and a decolorizer. Now this is the type of staining. So we have categorized staining techniques in two types, simple staining, differential staining. Simple staining is used of a single cell, single stain. Then the differential staining we use of two counter staining. Then simple staining, we further categorize them into two types, direct that is positive and indirect is negative. So differential staining, we have categorized them into two categories. First one is separation into two groups. So that one is gram staining and acid fast staining. So as you know, I have already given a question regarding acid fast staining. Then we have a visualization of structure, that is flagella stain, capsule stain, and spore stain. So then simple staining we have two types, direct and indirect. So direct is used to see the object and indirect is used to see the used to stain the background. Now you can see the picture of the direct staining. So the object is here stained, means the bacteria is stained. And this is the indirect stage, negative staining. So here the background is stained, but the bacteria is not stained. So this is the staining general technique. First we make a smear, then we air dry it, then we heat fix it, and then we stain it, and then we look it into the microscope. The next is importance of fixing the smear. Why we need to fix the smear? Because it can kill the microorganism and it cause the organism to adhere to the slide and it alter the organism so that they more rapidly accept the stain. Then the gram staining or differential staining. So in 80s, 1880s, 100, Christian Graham observed the general uh, staining technique of gram staining by using diiodide complex, and they rinsed it with alcohol. So this is the gram staining techniques. It will include positive and negative dye already. So this is the gram staining requirement. We need a crystal violet. So basically we need a primary stain, then we need a moderate stain, then we need a decolorizer, then we need a counter stain, and then we see it. So this is the procedure. So while we cover up the gram staining procedure, we can conclude it in the 4 stages. So this, as you can see in this picture very clearly, this is the gram positive bacteria's procedure and the gram negative bacteria. So this is the gram staining. First we have taken a slide and we have mixed all the gram positive and negative bacteria. Then we heat fixed it and then we add a primary stain. For example, we have taken crystal violet, then it is stained both the bacteria in the purple color. Then we take a moderate stain which fix their color. So we take iodine solution. Then we take a decolorizer, ethanol, which decolorize the gram negative bacteria. Then we take a saffron counter as a counter stain. So it will convert the decolorized gram negative bacteria into the saffron color, red color. So this is the picture of the gram positive and gram negative bacteria after gram staining. Now the acid fast bacteria that are mostly used for mycobacteria. So this is the picture view of Mycobacteria tuberculosis in sputum. So we have taken while doing the acid fast staining. Then we have capsule staining. Then we have a his method of capsule staining. The next is endospore staining. So for endospore staining, we need a Ziehl-Neelsen method. And we have other method also. It was method and Fluton stain techniques. Now this is the picture view of endospore staining. And now I hope you get your all the answer of these questions. For the reference purpose, you can go through the pharmaceutical microbiology of Blackwell scientific publication, Oxford. So this book is very helpful to study in detail. Then we have a Brown Publishers Oxford Microbiology 3rd edition by Prescott, LM, Hadley, GP and Callan, and you can also go through these books. Then we have third one is Pharmaceutical Microbiology by NK Jain. This book is very easily available, so you can go through this book also. Then we have some of the materials taken from the slides here, so you can see and check it from there. So as you can see, staining techniques are very important for checking or to know about the morphology of any bacteria, virus or any microorganism, so that we can know their structure, we can know their properties, their morphology. So staining techniques play a vital role for them. The most important thing in staining is we have to make a stain properly. We have to make a smear properly so that it can retain the dye properly and take give the color properly. And while we trying the staining techniques, we have also known that which for which bacteria we are staining it. According to that, we have to use the proper dye. Only then the color will be visible. Only then the bacteria will be visible. Then while we are doing heat fixation in smear fixation, we have to be very careful so that our slide will not broke out, and we have to allow the slide cooling for a proper time. And while we are doing methanol fixation of smear, so we have to use a proper amount of methanol and for the proper time so that our smear will not wash out properly, or our sample will not wash out. And we have to adjust our microscope properly also, so that we can check out all our samples clearly. It will be up to in the microscope should be very clean, and the lenses should be properly used. So this is all about your stain and staining techniques. I hope you are now able to differentiate between the stain and staining techniques, and you are now able to know about what and which bacteria we have to use, which kind of staining, and we have to apply which kind of method. So this is all about your stain and staining techniques. Thank you for today.